Welcome to the Creative Commons RX podcast. Before diving into this episode, I want to ensure we're all on the same page. This is for general informational purposes only and does not constitute the practice of medicine, nursing, or other professional healthcare services, including the giving of medical advice. No doctor-patient or pharmacist-patient relationship is formed. Using this information and the materials linked to this podcast is at the user's risk. The content on this podcast is not intended to substitute for professional medical advice, diagnosis, or treatment. Users should not disregard or delay in obtaining medical advice from any medical condition they have, and they should seek the assistance of their healthcare professionals for any such conditions. Clinical experts created the references, content, and clinical insight. NotebookLM, a Google AI tool, created the audio content, which I extensively reviewed before release. Finally, the host states that he takes all conflicts of interest seriously. Currently, there are no conflicts to disclose. For all of his disclosures and the companies he invests in or advises, he directs users to reach out independently, where he keeps an up-to-date and active list of all disclosures.
Okay, let's unpack this. We talk constantly, and rightly so, about the revolutionary potential of modern medicine. Things like cell and gene therapies, advanced biologics, and the promise of personalized cures.
Right, it's genuinely exciting stuff.
But there's an immense invisible framework that makes this revolution possible, and frankly, keeps it safe.
Absolutely.
This isn't just about laboratory hygiene. It's about controlling complex, dynamic, living systems that incorporate human cells, powerful viral vectors, and highly intricate biology.
That's the core tension we are diving into today. Our mission is to understand the integrated framework where biosafety, protecting the worker and the public environment, and quality, known as current good manufacturing practice or CGMP, are utterly interconnected.
They really go hand in hand then.
Completely. If you control the safety of your process, you control the quality of your product. It's almost inseparable in this field.
We're drawing on a comprehensive guide detailing everything from the definitions of risk to the meticulous validation protocols required to get these products to market.
A really thorough source, yeah.
And for you, the learner, the key takeaway driving all regulation in this space is a simple but profound statement. The process is the product. You need to understand how the moment-to-moment control of manufacturing fundamentally defines the safety and efficacy of the final dose.
Exactly. When dealing with living materials, the controls are everything. Let's start by establishing the paradigm shift that necessitated this level of stringency, because it highlights why traditional pharmaceutical rules simply, well, they just don't apply here.
Right. The pharmaceutical world used to be relatively simple.
Right.
We had small molecule drugs. Think of aspirin or ibuprofen.
Yeah, stuff we all know.
Our source notes that aspirin, for example, is composed of as few as 21 atoms, produced through reliable, predictable chemical synthesis.
Very straightforward.
If I manufacture aspirin in London or Shanghai, the final molecule is chemically identical. It is robust, and it is highly reproducible.
Dead simple, chemically speaking. But then you cross over into the realm of biologics.
Okay.
These are massive, complex molecules, proteins, peptides, DNA, RNA, often composed of over 25,000 atoms.
Wow, 25,000 atoms versus 21. That's a scale difference.
Huge difference. And crucially, they are derived from or produced by living organisms, engineered cells, bacteria, or yeast. This complexity introduces variability at a scale we simply didn't see before.
And at the absolute pinnacle of this complexity are cell and gene therapies, or CGT.
The cutting edge.
Cell therapy is the use of intact living cells, often immune cells, to treat a disease. Gene therapy involves modifying, replacing, or inactivating a gene, typically using a viral vector as a vehicle to deliver the genetic payload into the target cells.
These aren't just drugs in the old sense. They are advanced biologics, often delivered as a single, potentially curative dose. Pretty amazing potential.
Truly.
To round out the definitions, we must mention biosimilars.
Yes, biosimilars.
A biosimilar is a biological product that is highly similar to an approved reference biologic. Now, this is a critical distinction for you listening. They are not generics.
Okay, why not? Aren't they just copies?
Well, yes and no. A generic is an exact identical chemical copy. Think generic ibuprofen versus branded. Because biologics are so complex and variable, two manufacturers, even using the same gene sequence, will never produce an exact copy.
Never.
The living cell systems introduce tiny variations, so they must simply prove the copy has no clinically meaningful differences. It's a different standard.
That concept that a molecule produced by two different methods can't be identical brings us right back to the regulatory mantra. The process is the product. Why is this so central?
It boils down to inherent biological variability, specifically in protein structure.
Okay.
When you produce a complex therapeutic protein in a living cell culture, the cell isn't a simple factory, it's a dynamic organism. You inevitably see minor, natural, batch-to-batch variations.
Like what kind of variation?
The most common variation occurs in post-translational modifications, particularly glycosylation patterns.
Hold on, let's unpack glycosylation, because that term sounds intimidating, but it sounds really important based on what you're saying.
Well,
What are these sugar patterns, and why do subtle changes matter so much?
yeah, good question. Glycosylation is essentially the attachment of complex sugar structures to the protein after it has been synthesized by the cell's machinery.
Okay.
Think of these sugars as complex signaling flags or antennas on the surface of the protein.
Flags.
If the cell culture conditions change, even slightly, maybe in nutrient availability or temperature, the pattern of these sugar flags can change.
And why does this matter? What do the flags do?
These flags dictate how the drug is cleared from the body, how it folds into its correct 3D shape, and critically, how the patient's immune system recognizes it.
So even if the main protein part, the amino acid chain, is 100% correct,
Yeah.
a change in process, a new bioreactor, a switch in purification resin, or even just tweaking the growth media can subtly alter those sugar patterns.
Exactly.
And that subtle alteration can shift the entire safety and efficacy profile.
Precisely. The regulatory standard, therefore, demands that the product is defined not solely by the formula, but by the entire validated manufacturing process used to create it.
Right.
Any process change, no matter how small it seems, requires intensive comparability testing to prove the final product hasn't functionally changed.
Which makes sense why changing things after approval is so hard.
Incredibly costly and difficult. You risk basically voiding the original product definition in the eyes of the regulators.
The most extreme example of this paradigm must be autologous CAR-RT cell therapy. That's the ultimate scale-out model, right? Tell us how that puts immense pressure on SOPs.
Right. CAR-RT is highly personalized medicine. We harvest a patient's own T cells.
Okay, autologous meaning from the self.
Exactly. Then we genetically modify them using a viral vector, expand them in a clean room, grow lots more of them, and then infuse them back into that same patient.
So the starting material, the patient's T cells, is unique every single time.
Every single time. You are running a batch of one. You can't easily compare batch A from patient X to batch B from patient Y because the input cells are inherently different.
So how on earth do you validate a process like that? Doesn't the sheer variability of the biological starting material kind of negate traditional validation approaches?
That's the crux of the regulatory challenge. It's a really good point. Validation shifts entirely to proving the robustness and discipline of the manufacturing platform itself.
The platform, okay.
Flow systems, the equipment, the software, and crucially, the people running the standard operating procedures, the SOPs. The only constant is the process. And that process must be proven capable of reliably converting highly variable input into a safe and effective product.
Which puts an almost overwhelming burden on procedural discipline and those CGMP controls.
It really does. And this inherent variability leads to dramatically different risk focuses across these different categories of drugs.
How so?
Absolutely. For biosimilars, the critical risk is almost entirely clinical and internal to the patient. Immunogenicity.
Immunogenicity, meaning the body reacting against the drug.
Exactly. Subtle, invisible variations introduced by a slightly different manufacturing process can cause the patient's immune system to generate anti-drug antibodies, or ADAs.
Okay.
These ADAs can neutralize the drug, rendering it ineffective, or worse, cause severe adverse events.
And we don't have to look far for a cautionary tale. The historical Ypres case study illustrates this risk perfectly, doesn't it?
It really does. Ypres, which is erythropoietin, a drug to stimulate red blood cell production, was manufactured with a specific stabilizer, human serum albumin.
Right.
One manufacturer switched the formulation in the late 1990s, changing the stabilizer to something called polysorbate 80, and also changed the rubber stopper in the prefilled syringe.
Seems like minor changes.
On the surface, yes. But this combination of process changes led to trace amounts of compounds leaching from the stopper that subtly altered the protein structure. And suddenly, patients began developing pure red cell aplasia. A severe immune reaction where their bodies not only neutralized the drug but actually attacked their own red blood cell precursors.
So a subtle, invisible process change triggered a massive, potentially fatal autoimmune reaction. That is truly terrifying.
It is, and it reinforces why CGMP process control is absolutely everything for traditional biologics and biosimilars.
But for CGT, cell and gene therapy, the focus is dual, you said.
Yes. You still need CGMP for product sterility, obviously, but the safety focus dramatically expands to physical containment biosafety level protocols, or BSL.
Because you're handling active biological agents.
Exactly. We're handling replication-incompetent viral vectors, genetically modified cells that might carry potent genes like oncogenes. So the dual safety mandate is protecting the worker and the environment from the active agent, while simultaneously ensuring the product remains sterile for the patient. Two sides of the same coin. To manage these high-stakes hazards, everything in a modern biomanufacturing facility begins with a mandatory, systematic, and comprehensive risk assessment.
Not optional, then.
Absolutely not. It's driven by the Biosafety and Microbiological and Biomedical Laboratories Guidance, the BMBL. It's the foundational document.
And the assessment must clearly delineate two major components: agent hazards versus procedural hazards. Tell us about agent hazards first.
Agent hazards are intrinsic to the biological material itself. What is its pathogenicity? Can it cause disease? How severe? What's the infectious dose required to actually cause infection? What are the viable routes of transmission, inhalation, ingestion, skin contact? And crucially, does effective treatment exist if someone gets exposed?
Right.
For example, an agent might be highly pathogenic, but if its only known route of transmission is, say, direct blood contact, the risk profile is different than an agent that transmits easily via inhalation through aerosols.
Okay, that makes sense. So what about procedural hazards?
Procedural hazards, on the other hand, are the activities, the things you do in the lab that create the opportunity for exposure.
And what are the procedures that typically elevate risk the most? What are the big ones?
Anything that generates aerosols. That's the main concern.
Aerosols.
Yeah.
Like invisible sprays.
Exactly. Tiny droplets hanging in the air. Things like centrifuging without properly sealed rotors or buckets, vortexing samples vigorously, using sonicators, even vigorous pipetting, and critically any use of sharps like needles or scalpel.
Why sharp specifically?
Because they create an immediate rail past the skin barrier. A needle stick injury is a direct inoculation. Aerosols are invisible, they are highly mobile, and they are the #1 way an inhalation hazard agent finds its way to a worker. So the risk assessment combines these factors, the virulence of the agent plus the potential for procedural release, to determine the required BSL, the biosafety level.
This requires us to be extremely precise when we talk about risk group, RG, versus biosafety level, BSL. They sound similar, but they're different.
Very different, and it's a crucial distinction. The NIH guidelines classify infectious agents into 4 risk groups based on the hazard of the wild-type agent.
Wild-type meaning the original naturally occurring version.
Precisely. RG1 is benign, poses no disease risk. RG2 agents can cause human disease, but it's rarely serious and treatments are available. RG3 agents can cause serious or lethal disease, potentially via inhalation, but treatments may exist. RG4 is the highest serious lethal disease, high transmission risk, often no treatment available. Think Ebola.
Okay, so RG describes the inherent danger of the bug itself. What's BSL then?
BSL, biosafety level, describes the containment conditions, the engineering controls like specialized ventilation, the safety equipment like biosafety cabinets, and the specific practices and procedures required to safely handle that agent for the work being done.
Ah, okay. So RG defines the hazard, BSL defines the mitigation response, the safety measures you put in place.
Exactly right. This distinction allows facilities to work with highly engineered agents safely, even if their parent virus is high risk.
Can you elaborate on that lentiviral vector example you mentioned? Its parent is HIV, which is RG3, so technically requires BSL3. But often, work with replication-deficient lentiviral vectors happens at BSL2 or BSL2 plus right here. How does that work? Why not just insist on BSL3?
It's because the risk has been deliberately engineered out, or at least significantly reduced. Modern lentiviral vectors are cleverly designed. They're usually split into multiple plasmids, often 3 or 4 separate pieces of DNA that are introduced into packaging cells to produce the vector particle.
So it's not the whole virus genome in one piece.
No. Crucially, they lack the essential genes needed for the virus to replicate itself and spread indefinitely after the initial infection. The risk assessment demonstrates that unless something goes very wrong in the manufacturing, the resulting vector particle doesn't behave like the full wild-type HIV. Its risk profile is considered equivalent to an RG2 agent.
I see.
Therefore, BSL-2, or maybe BSL-2 with some enhancements, which we call BSL-2+, for large-scale work, is justified by the risk assessment. If we had to use full BSL-3 containment for every single step of producing and using these vectors, the cost and logistical complexity would be absolutely crippling, potentially stopping therapies from ever being developed.
That detailed assessment of risk reduction is really key then. But what are the specific hazards that keep biosafety professionals up at night when dealing specifically with cell and gene therapies?
Number one, by far the biggest concern is the potential generation of replication-competent virus, or RCV.
RCV, yeah.
We engineer these vectors, like lentivirus or retrovirus, to be non-replicating or replication deficient. But during the complex manufacturing process, where cells are co-transfected with those multiple plasmids you mentioned, there is a small but non-zero chance of a recombination event.
Recombination, like the pieces accidentally joining back together?
Essentially, yes. It's like a genetic cut-and-paste error where the vector manages to reassemble the missing genetic pieces it needs to replicate, potentially reverting to a fully functioning replicating virus similar to its wild-type parent.
So RCV means the gene therapy vector has essentially come back to life as a live, potentially contagious agent.
Precisely. And if RCV is detected in a batch of vector intended for patients, it is an absolute catastrophe.
What happened?
The lot is immediately destroyed. Production might be halted completely. It requires massive regulatory notification and a full-scale investigation into how it happened. It is why rigorous RCV testing is arguably the most critical quality control release test for any batch of viral vector.
Wow. Okay, what else? We also hear about the risk of insertional mutagenesis. This sounds mainly like a patient risk, but how does it translate into a theoretical occupational biosafety risk for workers?
Right. Insertional mutagenesis is primarily a patient safety concern. Vectors like lentiviruses are designed to integrate their genetic payload into the host cell's genome to provide a long-lasting therapeutic effect.
To make the fix permanent.
Exactly. However, that integration process is largely random. There's a chance the vector could insert itself in a bad spot, maybe activating a dormant oncogene, a cancer-causing gene, or inactivating a crucial tumor suppressor gene. If that happens, that specific cell could potentially lead to malignancy down the line for the patient.
Okay, that's the patient risk. What about the worker?
The theoretical worker risk stems from the possibility of chronic, long-term, low-level exposure to the vector. Imagine someone working with these vectors for years, maybe experiencing undetected small splashes or inhaling very low levels of aerosols day after day. If that vector gets into their own cells and integrates, there's a theoretical long-term risk that insertional mutagenesis could occur in the worker as well. It's considered a low probability, but it's a risk that needs to be managed through containment.
Makes sense. And we must also account for the transgene hazard itself, right? The danger isn't just the vector, but what the vector is carrying.
Absolutely critical point. The required containment level is dictated not just by the delivery vehicle, the vector, but by the biological activity of the cargo, the transgene it carries.
Give me an example.
If the viral vector is carrying a gene that codes for a highly potent toxin or a known powerful oncogene, the risk assessment will demand higher containment, maybe even BSL-3, regardless of the vector type. Conversely, if the vector is just carrying a simple reporter gene like green fluorescent protein, something used for tracking,
Something harmless.
then the risk assessment will likely conclude that standard BSL-2 is sufficient. The Institutional Biosafety Committee has to evaluate the biological function of the genetic payload itself.
Okay. Finally, there's the practical risk associated with patient handling after they receive the therapy, viral shedding.
Yes, this is a significant consideration, especially for in vivo gene therapies, where the vector is injected directly into the patient's body.
Right.
Patients who receive these treatments will often shed the vector for a period of time, days, weeks, sometimes longer in their bodily waste, like urine, feces, saliva, or sweat.
So the vector's coming out of them.
Yes. This creates a potential exposure risk for healthcare workers caring for them, for cleaning staff handling linens or waste, and even for family members at home. It mandates specific education and protocols for handling potentially contaminated materials and practicing good hygiene in the home or clinical setting after treatment.
And the related concern for the manufacturing workers themselves is accidental exposure causing seroconversion. What's that?
Seroconversion means developing antibodies against something. If a worker has a significant accidental exposure to a specific vector type, maybe a needle stick with adeno-associated virus, AAV, their immune system may recognize it as foreign and develop antibodies against that specific AAV serotype.
So their body now recognizes that AAV type.
Correct. They have now seroconverted. The consequence is, should that worker ever need a gene therapy themselves in the future that uses the same AAV vector type?
Which is quite likely, given how common AAV is.
Exactly. Their own immune system, primed by the occupational exposure, would immediately recognize and neutralize the therapeutic dose before it could even work effectively. They would be ineligible for that treatment. This is a profound, long-term occupational health concern that needs careful management and tracking.
Okay, so the result of this complex risk assessment is the assignment of a physical containment level. Let's start with BSL-2, which you identified earlier as the essential baseline for this entire industry.
That's right. BSL-2 is the fundamental level required for working with agents associated with human disease that pose a moderate potential hazard. These are the risk group 2 agents we talked about.
But why is it the baseline for cell and gene therapy?
It's simple. BSL-2 is the absolute minimum containment level required by regulations like OSHA's bloodborne pathogens standard for any work involving human-derived materials. That includes blood, serum, tissues, or established human cell lines.
Because of the risk of known human pathogens like hepatitis B or HIV being present.
Precisely. Since nearly all cell therapies involve handling human cells or blood products, BSL-2 practices and facilities become the mandatory starting point, regardless of the specific therapeutic agent being manipulated.
Okay. What are the defining characteristics of BSL-2 containment? What does it look like in practice?
It involves several layers. First, standard microbiological practices. Things like limited access to the lab, proper biohazard signage, policies against eating, drinking, or applying cosmetics in the lab. Hand washing is critical.
Basic lab safety stuff.
Yes, but formalized. Then there's the use of personal protective equipment, or PPE. Typically lab coats or gowns, gloves, and eye protection like safety glasses or face shields. And finally, the centerpiece of primary containment for procedures with aerosol potential at BSL-2 is the Class 2 Biosafety Cabinet, BSC.
The BSC. That sounds important. How does it work?
It is critically important. A Class 2 BSC is a ventilated cabinet that provides personnel, product, and environmental protection. It operates using carefully engineered airflows. Room air is drawn in through a front grill, preventing aerosols generated inside from escaping. Inside the cabinet, HEPA-filtered air flows downwards over the work surface, protecting the product from contamination. And the air exhausted from the cabinet is also HEPA-filtered before being released, protecting the environment.
You mentioned earlier the BSC is a perfect example of biosafety and CGMP conversions. How so?
Exactly. It serves two masters simultaneously. From the biosafety perspective, that inward airflow at the front opening protects the operator from breathing in any hazardous aerosols generated during manipulation.
Okay, worker protection.
Right. And from the CGMP, the quality perspective, that stream of HEPA-filtered air flowing down onto the work surface creates an ultra-clean environment, typically ISO Class 5 or Grade A, which protects the sensitive cell product from microbial or particulate contamination coming from the operator or the surrounding room air.
Product protection.
One piece of equipment doing both jobs. It's why proper use and maintenance of BSCs are so heavily emphasized in training and procedures.
Okay, let's move to the unofficial but apparently essential category, BFL2 plus C. You mentioned it's not in the official BMBL guidelines, yet it's widely used in the industry. Why did facilities sort of invent this pragmatic bridge between BSL2 and BSL3?
Yeah, it's an interesting case of practice evolving to meet a need. It really arose from a cost-benefit calculation driven by the scale of work and the nature of engineered vectors.
What do you mean?
Well, for certain activities, like producing large volumes of replication-incompetent lentiviral vectors, standard BSL-2 practices might feel insufficient to fully contain potential aerosols, especially if there was a large spill outside the BSC. But building and operating a full BSL-3 facility is incredibly expensive and introduces significant operational constraints, specialized training, complex airflow systems, rigorous entry-exit procedures.
So BSL-3 felt like overkill for reduced risk vector, but BSL-2 felt maybe not quite enough for large volumes.
Exactly. BSL-2 Plus became the institutional solution. It takes the foundation of BSL-2 practices and equipment and layers on specific facility design features and operational practices borrowed from BSL-3 without going all the way.
Okay, so what defines these BSL-2 plus enhancements? What are the key additions?
It usually starts with elevated PPE. Instead of just a standard lab coat, you might require solid front, back closing disposable gowns, double gloving, maybe N95 respirators, or even powered air purifying respirators, PPRs, for procedures with a high aerosol risk.
So more robust personal barriers.
Yes. But the key facility enhancement, the defining feature of BSL-2 plus WAY, is the requirement for inward directional airflow into the laboratory room itself.
Meaning the room is under negative pressure.
Correct. The laboratory must be maintained under negative air pressure relative to the adjacent corridor or non-lab areas. This means air always flows into the lab when the door is open and doesn't flow out.
Why is that so important?
It provides an additional layer of secondary containment. If there is a spill or release of aerosols outside the primary containment of the BSC, the negative room pressure helps ensure those aerosols are contained within the room envelope and pulled into the exhaust ventilation, preventing them from spreading out into hallways or adjacent clean office spaces. It's a critical engineering control borrowed from BSL-3.
Okay, that engineered negative pressure is the bridge. But BSL-3 represents a fundamental philosophical shift in containment, right? When we jump to BSL-3, we're no longer just relying on good practices in a cabinet. The facility itself becomes the primary containment system.
That's the crucial difference, absolutely. BSL-3 is specifically required for working with indigenous or exotic agents that cause serious or potentially lethal disease, primarily through the inhalation route. These are your risk group 3 agents. The entire facility is designed, built, and operated to contain these dangerous pathogens actively and passively.
Walk us through the key secondary barriers required for a certified BSL-3 space, starting with how you even get into the room.
Access control is paramount and strictly enforced. You typically enter a BSL-3 lab through a 2-door entry system, often called an anteroom or an airlock.
Like on a spaceship.
Sort of, yeah. The doors must be interlocked, either mechanically or electronically, so that you can never open both doors simultaneously. This maintains the air pressure difference, the containment barrier during entry and exit.
Okay, what else about the room itself?
The room construction is critical. All structural penetrations where pipes, conduits, electrical wiring, or ducts pass through the walls, floor, ceiling must be meticulously sealed using specialized sealants to ensure the room is airtight. This prevents any leakage of contaminated air into the surrounding building structure. Surfaces, walls, floors, ceilings, furniture must be smooth, seamless, non-porous, and resistant to the chemicals used for decontamination, allowing for easy and effective cleaning.
And the HVAC system? The heating, ventilation, and air conditioning? This is where the multi-million-dollar complexity truly resides, I gather. Explain the requirement for dedicated single-pass ventilation. That sounds important.
It's arguably the most critical engineering control in BSL-3. The HVAC system serving the BSL-3 suite must be completely independent and dedicated solely to that area. And critically, it must be a single-pass system, also called 100% exhaust.
Meaning the air isn't recirculated.
Correct. Fresh outside air is drawn in, conditioned, heated or cooled, supplied to the lab, used once, and then immediately exhausted directly back to the outside. Air is never recirculated back into the lab or anywhere else in the building.
Why is that non-negotiable?
It prevents any possibility of a contaminant released in the BSL-3 lab getting spread through the ventilation system to other parts of the facility, like offices or other labs. It also ensures the constant removal of any airborne contaminants generated within the lab. This system must also reliably maintain that measurable, constant negative pressure relative to the anteroom and the surrounding corridor, ensuring inward airflow at all times. This is often monitored with visible gauges or alarms, and the system undergoes rigorous testing, like pressure decay tests, to verify its integrity.
And where does all that exhausted air go when it leaves the facility? You can't just pump potentially contaminated air outside, can you?
No, absolutely not. Before being discharged outside, all exhaust air from a BSL-3 facility must pass through HEPA filters, high-efficiency particulate air filters.
HEPA filters. We hear that term a lot. How good are they?
They are incredibly efficient by design. Standard HEPA filters are rated to remove at least 99.97% of airborne particles that are 0.3 micrometers in diameter, which is the particle size considered most penetrating. They effectively trap bacteria, viruses, and other microbial agents.
Okay.
These HEPA filter banks are usually located near the exhaust fan, often in duplicate, redundant banks, for safety and maintenance. They must be tested and certified regularly, typically annually or semi-annually, using specialized techniques like DOP, dispersed oil particulate testing, or photometer scanning to ensure they haven't developed leaks and are still performing to specification. Furthermore, the final exhaust stack on the roof must be designed and positioned carefully away from public areas and building air intakes to ensure proper dispersion and prevent any possibility of the filtered air being accidentally drawn back into the building.
Operationally, inside the BSL-3 lab, things are also far more stringent than BSL-2 plus wear, right?
Oh, absolutely. The procedural requirements are much stricter. All open manipulations involving the infectious agent, not just those known to generate aerosols, must be conducted within a primary containment device, usually a Class 2 BSC, or sometimes an even higher containment device called a Class 3 BSC, which is a completely sealed glove box system.
Okay.
There is also the critical requirement for decontamination of all materials before they leave the lab. All waste generated inside use PPE, plasticware, culture plates, liquid waste must be decontaminated, usually by heat sterilization in a dedicated double-door pass-through autoclave that penetrates the lab wall. Items go in the contaminated side, are sterilized, and then removed from the clean side outside the lab. Nothing leaves without being decontaminated first.
Okay, moving from the physical structures like BSL-3 labs to the procedural ones that happen within them, or even in BSL-2. We established earlier that for these products, the process is the product.
Yep, the mantra.
This means that documentation and strict adherence to standard operating procedures, SOPs, is not just bureaucratic overhead. It's actually the ultimate quality and safety control measure. The regulatory mantra is uncompromising, you said. If an action was not documented, it did not happen.
That mantra really elevates the SOP from just a guidance document to a legally binding, auditable record of exactly what was done. The SOP life cycle in a CGMP environment is incredibly rigorously controlled.
Tell me about that life cycle. How does an SOP come to be and stay current?
Well, they are typically created based on data from process development, equipment validation studies, and comprehensive risk assessments. They need to be clearly written, unambiguous, and provide step-by-step instructions. But writing it is just the first step. It must then undergo formal review by subject matter experts, people who actually do the work or understand the science, and then critically receive mandatory documented approval from the Independent Quality Assurance QA unit before it can be officially implemented.
So QA's sign off as the gatekeeper. They hold the authority to say, yes, this procedure is validated and acceptable.
Exactly. QA ensures the SOP meets regulatory requirements, aligns with validated processes, and adequately controls risk. Once that QA approval is obtained, training on the current effective version of the SOP is mandatory for all relevant personnel, and that training must be documented.
Okay.
Then, the execution of the work itself, following that SOP precisely, is documented in real time, step by step, usually on specific forms called batch records or lab worksheets. Every action, every piece of data, every signature is recorded as it happens.
And they don't just sit on a shelf forever once written.
Absolutely not. SOPs are living documents. They must undergo periodic review, typically annually or biennially, to ensure they are still accurate and reflect current practice. And any revision, even seemingly minor changes like correcting a typo or clarifying a sentence, must go through a formal change control process.
Change control.
Yes. This process assesses the potential impact of the proposed change on the validated state of the process, product quality, and ultimately patient safety. Significant changes might require revalidation or additional studies before the revised SOP can be approved by QA and implemented. It's a very structured way to manage change.
This level of procedural rigor is absolutely necessary because, as you pointed out, unlike traditional drugs, cell and gene therapy products usually cannot undergo terminal sterilization at the end of the process. You can't just autoclave a bag of living CAR T cells or irradiate a vial of sensitive viral vectors.
Exactly. That would completely destroy the product. The living cells would die, the complex proteins or viral structures would be denatured or damaged. So the entire manufacturing process from start to finish must be conducted aseptically, meaning in a way that prevents microbial contamination from entering the product in the first place.
Okay, so if you can't sterilize it at the end, how do you prove that your aseptic process actually works? How do you convince regulators and yourself that you're consistently making a sterile product?
That's where the ultimate validation tool comes in. The aseptic process simulation, APS, which is universally known in the industry as a media fill.
Media fill. Okay, I've heard that term.
The media fill is the objective proof, the demonstration that your facility design, like the clean room classifications, your equipment, like filling lines or closed systems, your decontamination procedures, your detailed SOPs, and, most importantly, your trained personnel are collectively capable of producing a sterile product without introducing contamination time after time.
Describe the simulation. What does a media fill actually look like in practice? What happens?
You essentially mimic the entire complex manufacturing process exactly as it would be performed for an actual patient batch, from the very first sterile connection or vial opening right through to the final filling and sealing of the product container. But instead of using the actual expensive cells or viral vector,
Use something else.
yes, you use a sterile microbiological growth medium, typically tryptic soy broth, TSB, sometimes abbreviated as tryptic soy broth.
Why TSB specifically? What's special about it?
TSB is chosen because it's an excellent general-purpose, nutrient-rich broth designed to support the growth of a wide range of common aerobic bacteria and fungi, precisely the types of environmental microorganisms that are most likely to be introduced if there's a breach in aseptic technique by an operator or a failure in the cleanroom environment. If a stray bacterium lands in TSB, it will grow readily.
Got it. So you run the whole process with this nutrient broth. Do you try to make it easy or hard?
You absolutely must challenge the system under worst-case conditions. The simulation needs to represent the most complex and challenging version of your actual process. This means using the maximum number of personnel typically involved in the process, running it for the longest anticipated process duration, and deliberately simulating every planned aseptic manipulation and intervention that normally occurs.
Like what kind of intervention?
Things like making sterile connections between bags or tubing, taking in-process samples for testing, replacing filters, adjusting equipment settings, simulating operator breaks, even simulating potential non-routine events if possible. The goal is to stress the aseptic technique and environment as much or more than a normal production run.
Okay, so you've run the whole simulated process with TSB, filled hundreds or thousands of units, bags, vials, syringes, whatever the final container is. They are the crucial control units. What happens to them then?
Once the simulation is complete and all the media-filled units are sealed, they are meticulously inspected for any defects, leaks, or turbidity, and then they're sent for incubation. They are typically incubated for a minimum of 14 days.
14 days. Why so long?
To give even slow-growing microorganisms ample time to multiply to detectable levels. The incubation usually involves cycling between two different temperatures, typically around 20-25 degrees C for part of the time to encourage fungal growth, and around 30-35 degrees C for the other part to encourage bacterial growth.
And after 14 days you examine them, what are you looking for?
You're looking for any sign of microbial growth. Since the TSB is normally clear, any cloudiness or turbidity indicates that microorganisms got into that unit during the simulated process and have grown during incubation. Each unit is carefully examined visually under specific lighting conditions.
And what's the passing grade? How many contaminated units are acceptable?
This is where the standard is absolutely brutal, yet completely non-negotiable in the pharmaceutical and biotech industry, especially for aseptically produced products. The acceptance criterion is 0 contaminated units.
0.
Not even one.
0, nada, zilch. Even a single contaminated unit out of potentially thousands filled during the simulation constitutes a failure of the media fill.
Wow. What are the real-world implications of finding just one single cloudy vial after incubation? I mean, obviously you should figure out what went wrong, but beyond just investigating.
The implications are immediate and catastrophic for production. A single positive unit mandates an immediate halt to any further manufacturing of actual product using that process or line until the issue is resolved. It automatically triggers a full-scale, formal investigation, usually managed under the KPA, Corrective and Preventive Action System.
NPA, okay.
You must meticulously investigate to find the root cause. Was it a facility issue, like a problem with the cleanroom air handling? Was it an equipment failure, like a faulty seal on the filling machine? Or was it related to personnel, an error in aseptic technique by an operator?
And if it was an operator error?
If the investigation points to an operator failure, that person must be immediately removed from aseptic processing duties, thoroughly retrained, and then formally requalified, which often involves successfully participating in another media fill simulation under observation before they're allowed back into the clean room.
And what about validating the process initially or after a failure?
Initial validation of a new aseptic process or filling line typically requires 3 consecutive successful media fill runs, all with 0 contaminated units, to provide sufficient confidence. A single failure often means you have to discard the results of any previously successful runs in that series and start the 3-run validation campaign all over again from scratch after implementing corrective actions. This can cost enormous amounts of money in lost production time, investigation resources, and significant regulatory scrutiny. It is, without doubt, the highest stakes quality control test in the entire industry.
This really brings the entire discussion full circle back to that convergence of biosafety and CGMP quality we talked about earlier.
Absolutely. They are intrinsically linked, especially in aseptic processing. Think about that Class 2 biosafety cabinet. Again, the perfect physical representation of this convergence.
How so? Remind us.
A failure in aseptic technique by the operator inside the BSC, say, they accidentally reach over an open, sterile container instead of around it, is instantaneously both a potential biosafety incident, possibly exposing their sleeve or glove to the live vector or modified cells they're working with, and a CGMP deviation, potentially shedding particles or microorganisms from their gown or glove into the sterile product below.
One mistake, 2 types of failure.
Exactly. The protective air curtain might be breached, exposing the operator while simultaneously contaminating the product. The 2 risks, worker safety and product quality, are completely inseparable in that moment. Good aseptic technique protects both.
And for autologous CGT, like the CRT therapies, where the starting patient material is constantly changing and unique,
Yeah.
you mentioned you're not really validating the product itself batch to batch in the traditional sense, but validating the platform's unwavering consistency.
Precisely. Since the input is variable, the focus of validation shifts heavily towards proving the robustness, reliability, and control of the automated equipment, the closed system design, if used, and the associated SOPs. You validate that the manufacturing platform, the machine, the software, the single-use disposables, the defined process steps can consistently execute the complex cell harvest, genetic modification, washing, and expansion steps without relying heavily on error-prone human manual intervention in an open environment.
So the automation itself becomes a key control for both quality and safety.
Very much so. Closed, automated systems inherently minimize the opportunities for both microbial contamination of the product and operator exposure to the biological agents, thereby strengthening both CGMP compliance and biosafety simultaneously. We can install the most sophisticated multi-million-dollar engineered systems, state-of-the-art BSL-3 labs, fully closed automated processing units, but at the end of the day, they are all only as good as the people operating them and maintaining them.
The human factor.
Exactly. The human factor, the training, the discipline, the proficiency of the operator is arguably the single most critical active safety system in any clean room or containment lab. Machines follow instructions, people need understanding, skill, and vigilance.
Which implies that training has to go far beyond just sitting through a simple PowerPoint presentation once a year. What are the mandatory requirements for training and ensuring people stay proficient?
Training has to be comprehensive, competency-based, and ongoing. Initial training for anyone working in these environments covers a wide range of topics. The principles of microbiology and contamination control, specific SOPs for their job functions, proper clean room behavior, like slow, deliberate movements, gowning and degowning procedures, aseptic technique theory and practice, waste handling, emergency procedures, and the specifics of the biological agents they'll handle.
That sounds intense.
It is, and it must be followed by mandatory annual refresher training to reinforce key concepts and cover any updates. But just attending training isn't enough. Proficiency must be actively demonstrated and documented.
How do you demonstrate proficiency in something like aseptic technique?
For personnel working in aseptic processing areas like inside those BSCs, ongoing qualification typically requires successful participation in those routine media fill simulations we just discussed, usually at least annually, sometimes semi-annually. Their ability to perform complex aseptic manipulations over an extended period without causing contamination in the media units is direct proof of their skill. Failure means retraining and requalification.
And for high containment areas like BSL-3, you mentioned access is typically qualification-based as well, right? It's not just open to anyone who's had general training.
That's absolutely correct. Access to BSL-3 is not automatic.
No.
Personnel must typically undergo specific, rigorous BSL-3 training and then demonstrate through practical assessments observed by experienced staff or biosafety officers their competency in critical procedures before being granted independent access.
What kind of practical assessments?
Things like correctly performing the complex donning and doffing, gowning and degowning procedure specific to that facility, demonstrating proper use of the BSC, showing correct waste management and decontamination protocols, and knowing the emergency response procedures for spills or exposures within the BSL-3 lab. Proficiency might need to be re-verified periodically. If an operator is implicated in a significant contamination event, fails an environmental monitoring test, or even fails a routine gowning qualification observation, their clean room or BSL-3 access privileges are usually immediately suspended pending investigation and retraining.
Okay, let's pivot to decontamination, because you stressed earlier how critical the details are here, especially matching the disinfectant to the bug. For solid waste, autoclaving using steam under pressure is the preferred method for sterilization.
Yes. Autoclaving is the gold standard for sterilizing most solid waste from BSL-2 and BSL-3 labs because it's effective, reliable, and penetrates dense materials. But you can't just assume it's working. To ensure its efficacy, which must be total kill, the performance of each autoclave needs routine verification using biological indicators.
BIs.
Biological indicators. What are those? They are standardized preparations of microorganisms known to be highly resistant to the specific sterilization process being monitored. For steam autoclaving, the most common BI uses spores of a thermophilic, heat-loving bacterium called Geobacillus stereothermophilus.
Hardy little guys.
Extremely hardy.
Yeah.
Small vials or strips containing a known number of these spores are placed inside the autoclave load in the most challenging locations. Example, deep inside a dense waste bag. After the cycle, the BIs are incubated. If the spores have been killed, no growth, it provides biological proof that the autoclave cycle achieved sterilizing conditions. This verification is done routinely, often weekly, or even daily for critical applications.
Okay, so autoclaving for solids. But chemical disinfection for surfaces, equipment, spills, and liquid waste is where many laboratories, maybe those outside the pharma biotech CGMP world, get it wrong. You mentioned the critical distinction regarding disinfectant choice.
Yes, this is a point of frequent failure and misunderstanding, and it's absolutely critical for safety. You must choose and validate a disinfectant that is proven effective against the specific biological agents you are working with. Not all disinfectants kill all microbes equally.
And the key issue for many gene therapy vectors.
Many common viral vectors used in gene therapy, such as adenovirus or adeno-associated virus, AAV, are non-enveloped viruses.
Non-enveloped, meaning they lack that outer fatty layer.
Exactly. They lack the fragile, lipid-based outer envelope or shell that makes other viruses like influenza, herpes viruses, or HIV relatively easy to inactivate with simple detergents or alcohols. Non-enveloped viruses are structurally much tougher and more resistant to disinfectants.
And this leads to a crucial warning about commonly used cleaning products in labs.
Yes. The standard ubiquitous lab disinfectant 70% alcohol, ethanol or isopropanol, is completely ineffective at inactivating many non-enveloped viruses, including adenovirus and AAV.
So wiping down a spill of AAV vector with an alcohol wipe does basically nothing.
That's correct. You might physically remove some of it, but you haven't chemically inactivated the virus particles remaining on the surface. This is a huge potential safety gap if labs aren't aware of it.
So what should be used for these tougher viruses?
For these resistant non-enveloped viruses, the facility must rely on a stronger oxidizing disinfectant. The most common and recommended choice is typically a freshly prepared 1.0 dilution of standard household bleach.
Regular bleach.
Yes, but the details matter. Standard household bleach contains sodium hypochlorite, usually at 5–6%. A 1.0 dilution gives you a concentration greater than 0.5% sodium hypochlorite, which is effective. Freshly prepared is key because bleach solutions degrade over time, losing potency. And you also need sufficient contact time. The disinfectant needs to remain wet on the surface for a specified period, often 15 to 30 minutes, depending on the agent and surface, to ensure complete inactivation. Just spraying and immediately wiping dry won't work.
Those details, fresh dilution, correct concentration, sufficient contact time, are critical then. And the waste handling SOPs themselves are part of the containment strategy too.
Absolutely. They prevent cross-contamination and ensure waste is handled safely. All sharps must be immediately placed in designated, rigid, puncture-resistant sharps containers to prevent accidental needle sticks. Contaminated solid waste, like gloves and plasticware, must be collected in leak-proof biohazard bags, often red or orange, and usually placed inside rigid, leak-proof secondary containers with lids for transport.
And moving waste out.
Before any waste container, sharps, box, or bag is removed from the BSC or from the containment lab, especially BSL-3, its entire external surface must be meticulously decontaminated with the appropriate validated disinfectant. This ensures that no residual contamination is accidentally carried out on the outside of the container across the containment boundary.
Okay, finally, let's talk about emergency preparedness. Because even with the best training, equipment, and procedures, accidents can still happen. Let's look at spill response first. What if a significant spill of a high-risk vector occurs outside of a biosafety cabinet, say, onto the lab floor?
This is a serious scenario, potentially involving maximum aerosol generation and spread. The immediate protocol, which must be drilled into personnel, is safety first. Alert others nearby immediately, and then evacuate the affected area quickly and calmly. Close the door behind you to help contain the potential aerosol cloud within the room.
Then what? Just wait.
Yes. The area must remain unoccupied for a defined period, typically at least 30 minutes. This is a mandated minimum time frame based on air exchange rates, allowing time for the ventilation system to remove airborne particles and for heavier aerosols to settle onto surfaces. Rushing back in too soon increases inhalation risk.
And after 30 minutes,
Only personnel who are specifically trained in spill response procedures and equipped with the appropriate heightened level of PPE should re-enter. This would likely include fluid-resistant gowns, double gloves, eye face protection, and crucially, respiratory protection suitable for aerosols, such as an N95 respirator or often a PPR, especially for large spills of high-risk agents. They then follow a specific, validated spill decontamination procedure using absorbent materials and the appropriate disinfectant with the required contact time.
okay, that spills. What about direct personnel exposure, the really unfortunate accident, like a needle stick with a contaminated needle or a splash of vector suspension directly into the eye?
Immediate first aid is paramount to minimize absorption or infection. For a needle stick or cut from a contaminated sharp, the advice is generally not to squeeze the wound, but to allow it to bleed freely if possible, and then immediately wash the area thoroughly for at least 15 minutes with soap and water.
15 minutes, that's a long time.
Yes, it emphasizes thoroughness. If there's a splash to the eyes or other mucous membranes, like the mouth or nose, the area must be flushed immediately and continuously for at least 15 minutes using an emergency eye wash station or copious amounts of running water. Getting the material off or diluted as quickly as possible is key.
And following that immediate first aid, reporting must be instantaneous, you said earlier. Why the extreme urgency in reporting?
All exposures, even ones that seem minor or uncertain, must be reported immediately to the laboratory supervisor and to the designated occupational health provider or biosafety officer. There are several reasons for this urgency. First, proper documentation is required. Second, immediate medical evaluation might be necessary to assess the risk based on the specific agent involved. And third, and perhaps most critically for certain agents, immediate evaluation for post-exposure prophylaxis, PEP, might be required.
PEP, preventative treatment after exposure.
Exactly. Depending on the specific virus or biological agent involved, PP might involve taking specific antiviral medications, receiving an immunizing agent, or other medical interventions designed to prevent infection or disease from developing after the exposure has occurred. But for PEP to be most effective, it usually needs to be initiated as soon as absolutely possible after the exposure, ideally within 1 to 2 hours. Delaying reporting, even by a few hours, can dramatically reduce or completely negate the potential benefit of PP. That's why immediate reporting is a non-negotiable safety requirement.
This entire deep dive has really reinforced the sheer complexity involved in making these powerful living drugs safely and consistently. The lessons learned from things going wrong must constantly feed back into improving the system.
Indeed. And we see examples of this feedback loop. The E-Prix case, although tragic, taught the entire biopharmaceutical industry invaluable lessons about how even seemingly minor manufacturing changes, like a stabilizer or a container component, could have devastating, unforeseen immunogenicity consequences in patients. Vigilance around process change became paramount.
And conversely, we see positive evolution too.
Yes. The increasing adoption of advanced engineering controls, such as the automated, functionally closed processing systems, like the Clinimax Prodigy platform, often used in CAR-T manufacturing, demonstrate how technology can enhance both CGMP compliance and biosafety simultaneously. By enclosing the process and automating steps, you minimize the need for open manual manipulation by humans, thereby reducing opportunities for both microbial contamination and operator exposure.
But even with automation in high-tech labs, the grim reality of laboratory-acquired infections, or LAIs, persists. We still hear about them occasionally, even in sophisticated BSL-3 environments. It underscores the vulnerability of any system to what we might call procedural drift, or just simple human error.
That's exactly right. Procedural drift is insidious. It's the gradual, often unintentional deviation from established SOPs over time. Maybe it starts with a small shortcut, perhaps skipping one step in the lengthy gowning protocol because you're in a hurry, or propping open an anteroom door briefly, or not waiting the full contact time for a disinfectant. These tiny, seemingly insignificant human errors can accumulate until they eventually bypass or defeat the multi-million-dollar engineering controls and safety systems that were put in place.
Which brings us back to the human element.
It always does. It's why continuous, rigorous training, ongoing proficiency verification, and fostering a strong, non-punitive safety culture where people feel comfortable reporting near misses or potential problems without fear of blame are arguably the final, essential layers of protection. They compensate for the fact that humans, unlike machines, are not perfectly consistent.
Well, this has been a truly fascinating deep dive covering everything from the fundamental molecular complexity of these therapies and the regulatory consequences of their inherent variability, right down to the specific concentration of bleach required to clean a spill effectively, every human discipline required to execute these processes safely day in and day out.
It really is a field where the macro-level science meets the micro-level operational detail. To summarize the key takeaways for you listening, the safe and effective delivery of advanced biologics and cell and gene therapies hinges on a tightly integrated system. Biosafety containment levels, BSL, must be rigorously determined by systematic risk assessment of both the agent and the procedure. And product quality, CGMP, relies on meticulous adherence to validated processes, proven most stringently through the unforgiving standard of zero contamination in aseptic process simulations the media fills.
And if we try to connect this to the bigger picture, maybe the most important takeaway is that in this highly complex field, the human factor, a well-trained, technically proficient, consistently disciplined, and constantly vigilant workforce, is truly the most critical control measure. It often surpasses even the most sophisticated and expensive engineering systems in importance.
I absolutely agree. That's the ultimate challenge and responsibility. The complexity of these living therapies means that the science is not just about defining the cure itself, but also about meticulously defining, validating, and executing the complex human and technological system required to make that cure safely and effectively, often one unique patient batch at a time.
Which leaves us with a final provocative thought, perhaps. We've seen huge advances in automation and closed systems, driven partly by these safety and quality needs. What future advancements might eventually minimize or even eliminate the possibility of human procedural drift entirely? Could we reach a point where technology can fully solidify the integration of quality and safety, making the system foolproof? That's certainly a question worth exploring as this field continues its rapid evolution.
This is for general informational purposes only and does not constitute the practice of medicine, nursing, or other professional healthcare services, including the giving of medical advice. No doctor-patient or pharmacist-patient relationship is formed. Using this information and the materials linked to this podcast is at the user's risk. The content on this podcast is not intended to substitute for professional medical advice, diagnosis, or treatment. Users should not disregard or delay in obtaining medical advice from any medical condition they have, and they should seek the assistance of their healthcare professionals for any such conditions. Clinical experts created the references, content, and clinical insight. NotebookLM, a Google AI tool, created the audio content, which I extensively reviewed before release. Finally, the host states that he takes all conflicts of interest seriously. Currently, there are no conflicts to disclose. For all of his disclosures and the companies he invests in or advises, he directs users to reach out independently, where he keeps an up-to-date and active list of all disclosures.